Okay, hello everyone. So this time we're going to talk about sickle cell anemia versus malaria. Malaria is a disease. It's not a genetic disease. This disease is due to a parasite which is one cell big, so it's unicellular. You can see that this parasite is entering a red blood cell. This is the cell membrane of a red blood cell, and this is the parasite which will enter the red blood cell. This parasite is called Plasmodium. Well, it's from the Plasmodium group. So this parasite actually comes into a human's blood via a mosquito bite. So if a human is bitten by a mosquito and that the mosquito has this parasite, this one cell big parasite will then go into the liver cells, reproduce, then sometimes it can go to another mosquito, etc. The interesting thing is that it produces in liver cells and destroys some of the liver cells and destroys some of the red blood cells. Some of the symptoms: fever, tiredness, vomiting, yellow skin, seizure, coma, etc. So not a nice disease. But there is something which is quite striking and interesting. So we've got two maps here. So left map is the areas in the world where the malaria parasite is present. So it's mostly in Africa, a bit around the Mediterranean and India, and these southern parts of Asia. But what's interesting is that if we compare it to the HBS allele, so I'm going to explain what the HBS allele is. It's an allele which produces the mutated beta globin. So if we go back a few pages, the HBS allele is basically this DNA here of the mutant B globin. It's the allele which produces the mutant beta globin. Okay, so three possibilities. Either a person has two HBAs and the red blood cells are normal. Heterozygous HBA, HBS, some of them have the mutant beta globin, and HBS, all the red blood cells have this mutant beta globin. Okay, so there's something striking, is that there are areas where this HBS allele frequency is higher. We could imagine that this frequency should be roughly the same everywhere in the world. However, it seems to match quite well the areas where malaria is present. It's not pitch perfect, but it is quite telling that the areas where malaria is present, the frequency of HBS is normally high. So scientists have been wondering how come, and there are a few explanations. Three possible genotypes: HBA, 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 HBS, two HBSs. So what we can have a look at is this research here. So researchers have compared the relative intraparasitic MI of 451 levels. So what does that mean? human cells can produce this microRNA. So these human cells can produce microRNAs 451. It turns out that people who have the HBS allele produce more. So the fact that they have this allele, HBS, that means they produce more of this microRNA 451. So people with both alleles, so homozygous for the HBS allele, they produce 1. HBA and HBS, so heterozygous, they produce a little bit, they produce roughly half. And those who don't have HBS allele, they produce much less. What's interesting is to compare it to the relative parasite growth rate. So people with HBAA, the growth rate of the plasmodiums, the parasite responsible for malaria, is very high, 5. Those with HBSS only 1, and those with HBA and HBS 2. So this is the key to the explanations. So people who have HBAA, they don't have sickle cell anemia. However, the growth rate of parasites is 5 times higher than those with HBSS. So those with HBSS, they have a severe form of anemia. However, they don't have they don't suffer from malaria so much. And those of HBAS, they have a similar growth rate to HBSS. So this is what I just said. So case 1, we can sum up that they don't have anemia, but they suffer more from malaria. HBSS, they have a severe form of anemia, but they do not suffer much from malaria. So it turns out that the heterozygotes, HBA, HBS, which can also be called people with the sickle cell trait, when they have one allele of each, so they have sickle-shaped red blood cells. So they suffer a little bit from anemia. And a little bit from malaria. So this intermediate phenotype is telling us that it's actually better to have a little bit of anemia rather than having severe malaria. So basically, the fact that the allele is so high, it's because this is explained by natural selection. People who have a little bit of anemia and a little people who have HBS and HBA, when there is malaria, they will survive more than people who have not sickle cell anemia. So it's better when there is malaria to have this phenotype. It's basically better to suffer a little bit from anemia, but not to suffer from the severe forms of malaria. HBS, HBS, they suffer greatly from anemia, and HBA, HBA, when there is malaria, they suffer a lot from malaria, so these phenotypes tend to disappear, these two ones, and the heterozygotes, they survive more. So gives a natural selective advantage in areas where malaria is present. Okay, so that's for malaria versus anemia. Goodbye.